Hi everyone, welcome back to Useful Genetics. This is lecture 9b. We're going to extend our discussion of heritability in the previous lecture to how can we actually estimate heritability. We'll talk about how to estimate it in breeding studies of plants and animals, a way to estimate it for ecological and evolutionary studies, and how to estimate it in humans when we can't actually control what humans do. So we need to find ways to separate the effects of genes and environment when we don't know which genes matter or which aspects of the environment matter. There are several ways we can do this. One is to measure a population's response to a single generation of defined artificial selection. Another is to remove the environmental variation completely, as much as possible. And the third is to compare the observed resemblance between relatives whose phenotypes we've examined to their predicted genetic relatedness. And I'll discuss all three of these. So first, for breeding studies, it's very important to know what the heritability is before doing the work. So one way to do that is to do one generation of the work and very carefully measure the response to selection. So imagine you have, say, a large breeding colony of pigs and you want to select for greater weight at 6 months of age. And so you take your population and you carefully measure the distribution of weights at 6 months of age. And then you breed only from the pigs who have high weights at 6 months. These are the desirable pigs. If you find that in the next generation, when you again carefully measure the distribution of weights at 6 months, that their distribution is like that of the whole parent colony, you'd say, oh, heritability is very low. Maybe it's 0. We had better work on improving the nutritional status of the pigs instead. If you find that heritability is extremely high, you see that the offspring population has the same distribution as their parents do, then you would conclude that heritability was very high and the breeding program was likely to be very successful. More likely what you find is that there is some heritability for height, that by selecting high weight parents, you produced offspring whose weights were definitely higher than the original full population, not as high as their parents. That tells you that heritability is somewhere in the middle. Now, in ecology studies, you don't have defined populations, but it's possible to do a different kind of experiment. You can take subpopulations, say, from different environments with different phenotypes and find out to what extent the phenotypic differences are due to genetic differences rather than to environmental differences by transferring a population to a controlled common environment. Here we've taken seeds from plants whose phenotypes were tall in one environment, short in another, and we're growing them in a common greenhouse. In plant biology, this is often called a common garden experiment. And it lets you discover how much of the phenotypic difference is due to genetic differences. Now, in humans, we can't do these kinds of experiments. Instead, we have to work with whatever information normal human behavior has given us. And so an excellent strategy that's very widely used is to compare the phenotypic similarities of relatives to the similarity that would be expected if the trait were perfectly heritable. And you can do this because we can calculate how genetically similar we expect known relatives to be. We'll talk more about this in a later lecture in this module. But for now, here's some basic information. For identical twins, the expected relatedness is 100%. They're genetically identical. And the similarity of their environment is quite high. We can contrast identical twins with fraternal twins. They have only 50% of their genes in common, but their environment that they grow up in is very similar to the level of similarity between the environments of fraternal twins is very high, like the level of similarity of the environments of identical twins. That lets us see the effect of different levels of relatedness with the environment as constant as we can keep it. Another way to do this kind of study would be to compare, say, fraternal twins, siblings, and parent and child, all of which are expected to be 50% identical. So fraternal twins are 50% identical, as are siblings, as are a parent and a child. But the levels of similarity in their environments are quite different. Siblings, because they're different ages, will have moderately different experiences within the family, and parents and child typically have quite a lot of difference in their environments. And these kinds of experiments can let researchers fish out the genetic components and the environmental components of phenotypic traits that they're interested in. So we've talked about different ways that heritability can be estimated, even though the genes and environmental factors responsible are completely unknown. In breeding projects, it's wise to do one generation of breeding with selection and carefully evaluate how much effect the selection has. In ecological and evolutionary studies, you can rear populations that have different phenotypes in a common environment, ideally one that you can control. And for human studies, the usual strategy is to carefully compare the level of phenotypic relatedness with the predicted level of genetic relatedness. Coming up next, I'm going to go through a couple of examples of studies showing the heritability of human traits. I hope to see you there.